All right, the topic that we're going to discuss today is sex-linked inheritance. As we know, in the case of human beings, there are 23 pairs of chromosomes, 23 pairs or 46 number of chromosomes. Out of that, 23 pairs of chromosomes. That means each and every cell of our body has 23 pairs of chromosomes. Out of that, the 22 pairs are called autosomes. This is what we need to remember. 22 pairs are called autosomes. And the 23rd pair, or we can say one pair, the last pair, the 23rd pair, one pair is the sex chromosomes. Sex chromosome. Now, we need to remember there are far number of genes than the number of chromosomes that is present in each and every cell of our body. We have just 23 pairs, but the number of genes that control as XY. And in the case of woman, it is represented as XX. So from this, what we understand here is the 23rd pair in the case of men is heterogametic. That means there are two different type of chromosomes, X and Y. In the case of woman, what we see here is XX, so they are homogametic. Both the chromosomes are XX, that is in the case of woman. Now, the genes that are located on the autosomes, that is the 22 pairs of chromosomes, genes that is located on number one, autosomes. We have just talked about the 22 pairs. These 22 pairs of chromosomes that are called the autosomes, the genes that are present on them are called autosomal genes. So basically, these autosomal genes are same for both male and female. Almost same for both male and female. Now the genes located on the sex chromosome, or we can say X chromosome, is the sex-linked genes. So in this particular segment, this is what we are going to talk about, the sex-linked genes that is present on the X chromosome. The genes that are located on Y chromosome are called Hollandric genes. Hollandric genes. These are basically present only on Y chromosomes, and they are basically inactive, genetically inactive. They are the Y chromosomes, the genes located on the Y chromosome are basically inactive, and these genes mostly code for, in the case of men, it is hairy pinna and SRY genes. SRY genes, we have talked about this previously. SRY genes means sex determining region of Y chromosome. SRY gene means sex determining region of Y chromosomes. So this is basically the genes that is located on the autosomes and the X chromosomes. On the autosomes, they are called autosomal genes. On the genes that are located on X chromosomes are called sex-linked genes, and genes that are located on Y chromosome are called Hollandric genes. In the case of mammals, the female are homogametic. That is what we have just talked about. The female is XX, so they are homogametic. Both the X are of same type. Whereas the male are heterogametic. That means they have X and Y chromosome. Now let's talk about inheritance of gene. Inheritance of genes on X chromosome. That is the sex-linked genes. The inheritance of how this is inherited. Inheritance of X-linked genes. Inheritance of X-linked genes. That means the genes which are present on the X chromosome. Inheritance of genes of the X chromosome, or the genes that is located on the X chromosome. We are going to talk about the inheritance of this. The phenotypic expression of genes located on X are known as the sex-linked inheritance. The phenotypic expression, that means whatever genes that we have in our body, they express themselves. They express themselves by carrying out certain activities, certain functions, certain cellular functions, or we can say the outward projection, outward features of the organism. So the phenotypic expression of the genes located on the X chromosomes are known as the sex-linked inheritance. So we are basically talking about those genes that are located on the X chromosome. Autosomal genes are genes expressed almost same both in the case of male and female. I have already talked about autosomal genes are almost the same in both male and female cases. Examples of autosomal genes are the color of the eye, the earlobes, whether it is detached or attached, baldness, that means loss of hair, tongue rolling, the ability that is present in some people to roll the tongue like this, it is called tongue rolling. This is also an autosomal gene. And the skin color and various other features. So autosomal genes, these. Now the sex-linked genes, or the sex-linked genes that are responsible for different characteristics in our body. Basically what we are going to discuss here in this particular chapter is two conditions, the hemophilia and color blindness. These are X-linked or the sex-linked genes that are present on the X chromosome. And Y-linked genes we have already talked about. Pinna, hairy pinna, that means hairs on the pinna, and SRY gene. We have already talked about this. However, the inheritance of sex-linked genes is an exception to Mendel's law. We have already talked about Mendel's law, how it is transmitted from one generation to another, and it follows certain rules. But this, the sex-linked inheritance, does not go along with the Mendelian rules. That is why it is an exception to the Mendel's law of inheritance. X-linked or sex-linked genes is transmitted to daughters only. X-linked genes are transmitted from father to the daughters only. From fathers to the daughters. Whereas in the case of son, they only get it from mother. So what we see here, father can transmit the characteristics to the daughter, but he cannot transmit the same characteristics to the son. Why? Because father has X and Y. Only thing he can transmit to the daughter is this X. If he transmits Y, then it becomes automatically a male. So the father can contribute this X for the formation of the daughter, and father can contribute Y for the formation of son. So what we are talking about here is the father, if this has certain sex-linked characters in this X, this cannot be to the son. He can transmit only to the daughter. Similarly, the mother can only transmit the X to the son. The sex-linked gene to the son. So what we see here, there is a cross from father to the daughter and from mother to the son. So what we see here is, this is called a crisscross inheritance, as there is change of sex in the next generation. The character that is present in the mother is transmitted to the son in the next generation. The character that is present in the father is transmitted to the daughter in the next generation. So what we see here in sex-linked inheritance, it shows a crisscross inheritance. This is very important, crisscross inheritance, because there is change of sex in the next generation. The character changes the position from male to female and from female to male in the next generation. That is why this is called crisscross inheritance. Sex-linked inheritance or color blindness. The first that we are going to talk about in the sex-linked inheritance is the color blindness. Remember, color blindness is a recessive trait. Color blindness is a recessive trait. I'm sure you remember what is recessive and what is dominant. Dominant, even if one dominant gene is present, that is sufficient to show to express itself. But for a recessive, it is possible only if it is in homozygous condition or when the gene is double in number. So color blindness being a recessive trait, it can show itself in the case of female only when it is in double in number. But it can show itself in the case of male even if it is single. Okay. Now as far as color blindness is concerned, a brief history about color blindness. John Dalton was color blind, but he did not understand the reason why. He was very curious to know why he was color blind, but he could not find out while he was living. So what he did was he contributed his eyes, he donated his eyes for a research to find out what was the cause of color blindness. John Dalton. And in 1944, his eye after his death was extracted and was dissected and a DNA analysis was done. And it was found out that his retina lacked one of the three pigments. The three pigments which are known as the photopigments. The three pigments known as photopigments. These three pigments which are known as photopigments, how do they work? They absorb the particular wavelength of light. Basically it is blue, red and green. The three primary colors with the help of which a person can distinguish different colors. It's somewhat like a painter mixing the paint, to bring out different type of shades. So these three pigments in unison work together to express or to find out what are the colors. They absorb, they capture. So in the case of Dalton, the cone cells, basically eye has two type of cells, the rod cells and the cone cells. The cone cells are responsible for colored vision, and they have these pigments that are present, the photopigments which are present, which enables to capture the incoming length of the light. In the case of John Dalton, one of the pigment was not being able to be captured by the retina, the cone cells of the retina. That is why he was colorblind. So that was the first case when first experiment to find out the cause of color blindness. Each photopigment consists of vitamin A. Remember that these photopigments, each of them consist of vitamin A, which produces a substance called retinol. It produces a substance called retinol, and there is another portion in the pigment which is called opsin. This is basically a protein. So where does the defect lie? Not with vitamin A, but with this opsin protein. The gene that is responsible for production of this protein becomes defective, due to which a person becomes colorblind. So The photopigments consist of two portions, vitamin A, that is called retinol, and a protein part which is called the opsin. And the defect lies in this opsin, which is basically a protein, because the X chromosome, which has the gene for the photopigment, is somehow has become defective, so it cannot produce that particular pigment in the cone cells of the eye. Alright, now let's talk about the photopigments. The three photopigments, the first is chlorolib. Chlorolib is basically responsible for absorption of green color. Basically responsible for capture of green color, absorption of green color. Second is erythrolib for red color. And the third is cyanolib for blue color. Cyanolib for blue color. Out of these three, these two are responsible for color blindness due to the defect of the gene in the X chromosome. But for the blue, the defect of blue, the color blindness for blue, is a rare case. This is found in the autosomal, it is an autosomal disorder due to the mutation of chromosome on the chromosome number 7. So this is a rare case. Blue color blindness is rare case. It is an autosomal disorder and it is due to the mutation of chromosome number 7. Chlorable, the name for this color blindness is protanopia. This is called protanopia. The name of the disease, that is color blindness for green color, is protanopia. For red color is deuteropia, and blue color blindness. So these are the different names and the different pigments that are responsible for causing color blindness. Blue, red and green. Blue, red and green, out of which blue is an autosomal disorder, but the green and red color blindness is a sex-linked disorder, which is basically due to the mutation or change in the X chromosome. Now, genes for color blindness is, how do we represent them? When we have to devise a ray diagram to find out, analyze this case. The gene for color blindness is represented as small c, and gene for normal vision, or we can say wild type. Wild type or normal vision means it is represented as C plus. C, only C, small c is for color blindness, and small c plus with a plus sign is for normal or the wild type vision. Okay, that is how it is represented. Now let's make a cross. A colorblind male is crossed with a normal female. Colorblind male is crossed with a normal female. You got to understand these are the symbols. This is for male and this is for female. So colorblind male, C, and other is Y. You represented it like this. In the case of female, it is C plus and C plus. This is a normal female. Okay? When these parental generations are crossed, the gametes that is produced, gametes in this particular case is C and the Y chromosome, and in this case is only one type because it is homogametic, so only one type of gamete that is produced. So in the F1 generation, what we see here is this combines with this to form C and C plus, and this combines with this to form C plus, and so what we see here is in the case of F1 generation, the C plus, the C is transmitted from the father to the daughter. So this becomes the daughter who is a carrier. She is not colorblind. Why? Because because color blindness is a recessive trait. So since she has a normal gene here, it suppresses the effect of the recessive trait. That is why we have a daughter who is a carrier. In the case of male, it is normal son. Normal son is produced. This is in the case of F1 generation. Okay? So what we see here is the color blindness from the father is transmitted to the daughter, not to the son. We are coming close to understand the crisscross inheritance here. Now, when the F1 generation, the members of the F1 generation were crossed or selfed, as we have talked about in the case of Mendel studies. When the F1 generation is selfed or crossed amongst themselves, then we have another scenario here. A carrier female is crossed with a normal male. Carrier female is crossed with a normal male. Now what happens here is, in this case, the gametes are again of two types of gametes. Here obviously we have two types of gametes because one is X and the other is Y. This is the case when we cross the F1 generation amongst themselves or selfed. Okay, so the result of the F2 generation will be something like this. This combines with this to produce another carrier female. This combines with this to produce a colorblind male. This combines with this to produce a normal female, and this combines with this to produce a normal male. So what you see here in this particular case is, now the mother has transmitted the defective gene, that is the colorblind gene, to her son only, not to her daughters. Daughter, she was already a carrier, so it is same as the mother. Okay, so daughter is carrier, whereas the victim is the son. So this is a case of crisscross inheritance. In the F1 generation, we saw that the father transmitted the defective gene to the daughter, who became a carrier. Now in the F2 generation, the mother of the F1 generation transmitted that to her son in the F2 generation. So what we see here, in every step there is a change of sex by the gene. This is called the crisscross inheritance. Hence we see that the father transmits the color blindness to the daughter, in the F1 generation, and in the F2 generation, the carrier mother transmits to her son in the F2 generation. Rare in case of female, in the case of female, a female becomes colorblind only if she is homozygous for the recessive trait, which is a rare, pretty rare case where the female, what we can basically understand is when both of these are recessive trait, then only the female becomes colorblind. Okay, so this is what is crisscross inheritance and transmission of the colorblind gene. So basically with this we find out that this particular trait is the X-linked. Now we go on to the other case study, that is another sex-linked disease which is called hemophilia. Hemophilia, which is also known as the bleeder's disease. This is also known as the bleeder's disease. As the name suggests, bleeder's disease, because a person dies due to excessive loss of blood, because the blood fails to clot. And this particular disease was first detected in the royal family of England. That is why it is also known as blue blood disease. This is also known as blue blood disease. This is basically a congenital abnormality in man. This is a congenital abnormality in man, in which case the blood fails to clot. And that is why a person who is suddenly injured will die due to excessive loss of blood if blood transfusion is not the immediate possibility. This particular disease is more common in men because this is also a recessive trait, and in the case of men it is more common because in the case of men there is only one X, so it is hemizygous state, hemizygous condition where only one X is present. That means the gene that is in question is only present once. That is why it is more prevalent in the case of men. Now this hemophilia is of two types, hemophilia A and hemophilia B. The hemophilia A is more classical, or this was the one that was detected in the case of the royal family of England. This is a classical or the royal hemophilia due to deficiency of anti-hemophilic factor or factor 8. This is due to deficiency of hemophilia A is basically due to deficiency of anti-hemophilic factor or we can say factor 8. Anti-hemophilic factor or factor 8. This is basically a more you can say less aggressive type of hemophilia, and most of the people, 80% of the population suffers from this type of hemophilia, anti-hemophilic factor or deficiency of anti-hemophilic factor or factor 8. B, hemophilia B is due to the deficiency of plasma thromboplastin factor. It is due to deficiency of plasma thromboplastin or factor 9. This is basically a defect in the protein that is required for the clotting of blood. This is basically the defect in the protein that is required for the clotting of blood. This is almost only 20% of the population suffers from this type of hemophilia. Again, like in the case of color blindness, the wild type is represented as H plus, the hemophilic, that is wild type or the normal type, and hemophilic type is represented as H. Hemophilic type is represented, the hemophilic gene is represented as H, and the normal is H plus. Now in this case, we draw the ray diagram of Queen Victoria and Prince Albert. Queen Victoria and Prince Albert. Queen Victoria is a carrier female. Queen Victoria is marries Prince Albert, who is a normal as far as hemophilia is concerned, normal male. In this case, there are two types of gametes. Here also two types of gametes. Two types of gametes. When this is crossed with this produces a normal daughter, normal son, carrier daughter, and a hemophilic male. Hemophilic male. So here also what we see, there is a crisscross inheritance, like in the case of the color blindness. Now, what is the difference between sex linkage and autosomal linkage? Sex linkage located on X chromosome. Autosomal linkage is present on the autosomes, that is the 22 pairs of chromosomes. Sex linkage located on X chromosome. Autosomal linkage is present on the autosomes, that is the 22 pairs of chromosomes. Sex linkage shows a bias as far as its transmission. The female can transmit it only to the male member, and the male can transmit it only to the female member. So it shows a sex bias. But in the case of autosomes, it is autosomal linkage, it is not seen. In the case of sex linkage, it is a crisscross inheritance, whereas in the case of autosomal linkage, it does not follow that. So this is basically, you can say, the sex-linked inheritance. In the next segment, we are going to talk about mutations and various other topics. All right? Thank you.